Greetings everyone. Welcome back to our ongoing exploration of the excretory system in week 7 of Introductory Animal Physiology. In today's session, countercurrent mechanism, RAAS, and diuretics will delve into the mechanisms by which the kidney efficiently concentrates urine and maintains electrolyte balance. We will discuss the countercurrent mechanism, a crucial process that allows the kidney to conserve water. We will also examine the renin-angiotensin-aldosterone system, the RAS, and its regulatory role in blood pressure and fluid balance, followed by an overview of diuretics and their physiological effects. These topics will help you understand how the kidneys adapt and regulate to maintain homeostasis, especially in response to varying physiological conditions. Let's dive in and explore these fascinating systems together. So, traditionally, the countercurrent system will have two parts. One is called as countercurrent multiplier, another one is countercurrent exchanger. This countercurrent multiplier involves a structure called loop of Henle, whereas the vasa recta is involved in the countercurrent exchanger system, where the peritubular capillary play a major role. Now, both the countercurrent multiplier as well as the exchanger, this why it is there? Just to establish an osmotic gradient throughout this region and to maintain the homeostasis. So, why it is called as a countercurrent? Any system can be called as a countercurrent where the flow should be in an opposite direction. Which flow? The fluid flow should be in opposite direction, that's in parallel, as well as the adjacent tubes. Like this is one tube and this is another tube where one will flow in this direction and the another will flow on the opposite direction. Then it will be called as counter current. Then here you can see the loop of Henle as well as the vasa recta structure, where it involves the flow in an opposite as well as in the parallel structure. That is why it is called as countercurrent mechanism. The first part of the countercurrent mechanism is that countercurrent multiplier. Why it is called as multiplier system? Because the normal osmolarity of the plasma is 300 milliosmoles per kg of water, whereas this multiplier system is there just to concentrate at its maximum for the maximum reabsorption of the water as well as the solutes. So that is the main function of the countercurrent multiplier. So the countercurrent multiplier, how it's happening, the function. So I'll just draw the loop of Henle here, right? So the structures involved in the countercurrent multiplier system is that ascending limb of loop of Henle and the descending limb of loop of Henle. So the ascending limb having two parts, one is thin and another one is thick, as you know, and the descending limb of loop of Henle. Descending means it will come from higher to the lower, ascending means it is from the lower to the higher, right? That you know. So the descending limb, always remember this law in the countercurrent mechanism, it is impermeable to, this is impermeable to water, it is impermeable to solute. The descending limb is impermeable to solute, whereas the ascending limb, which is impermeable to water. So here which one is permeable? The water is permeable, whereas here which one is permeable? The solute is permeable, whereas water is non-permeable. Remember, this is one important law, rule you should remember before studying the countercurrent multiplier system. Now, the normal osmolarity, you know how much it is? 300 milliosmoles. So let us see here 300, 300, 300. Everywhere we will write 300. Now I will tell you how this countercurrent mechanism happens. So this is descending limb and this is ascending limb. Now everywhere it is 300 because that is the normal osmolality of the plasma, right? Now what I told, the descending limb is permeable to water. So what is happening? How will I understand this one? Let us start with very simple example. I'll give you 10% solution. 10% solution means what? 10 grams of any compound in 100 ml of water. Then you can call it as a 10% solution, right? Now we have a beaker, right? 10% solution in it. That means 10 solutes you have taken where 90 ml of water you have added and you made it up to 100 ml. So that is 10% solution. You can call it as 10%. Now what will happen if I removed 50 ml of water from this? What I am doing? Removing 50 ml of water from this beaker. So same amount of solute, that means the 10 solute particles are there, but I am removing 50 ml of water. So as a result, what happens? The water level reduces, but the same amount of solute is present. Now what is the concentration of this solution? Whether the concentration is increased or decreased, that means it will become an hypertonic solution. Before it was not, but now it is hypertonic. That means solute concentration is fixed, but the solvent, that means the water level is decreasing. So that due to that reason the concentration increases. Okay, the same thing applies even here also in the nephron, right? In the descending limb of loop of Henle, what is happening? The water is moves from the tubular fluid section to the interstitial fluid. Okay, this is the interstitial fluid section, so it is just moving from this. So what will happen? Water is moving, so as a result this 300 it will become 400. How it become 400? I gave you the example here. 10% it becomes, the concentration increases. No, same way here also the 300 become 400 because the water moves out and the solute stays as it is. So due to that reason the concentration rises. So for example, now again it become 400 here, right? Now this is of the descending limb. Let us move to the ascending limb now. Now ascending limb, it is having 300, right? Now here what happens in this 300, from it is permeable to the solutes but not to the water. So that means what? I am removing the solute particles from this, okay, not the water. The same way here, only the solute particle is moving. When the solute particle is moving, then what will happen to the concentration? The concentration reduces, okay, from the normal level. So that means from 300 the solute will move this side, so it will become 200. So that is why you might have seen in some book, the first and foremost step is that on the descending side you will find there is an increment of 100, whereas on the ascending side you will find there is a decrement of 100, okay, and you will find the difference between these two limbs as a difference is 200, okay. That is the most important thing, right? So now what happens? We have 400 here, 400 here, here we have 200. Now next what is happening is that again from here the water is moving from the descending limb side. So that means what happens? This 400 will become 500, correct? Then let us move on to the ascending limb side where the solute will move. That means this 400 will come here. So as a result what will happen? From here again the solutes will move. This 400 will become 300, right? So that means what? Again the difference is 200. So like this it will keep on concentrating, okay, it will keep on concentrating. That means the movement of the water from the descending limb and movement of the solutes from the ascending limb. So this where it is going? It is going to the interstitial fluid, remember. This is very, very important. Just understand so that the counter current multiplier system will not be a problem for you at any level. So this is how it will keep on increasing from 500, it will keep on increasing to 1200. Whereas in dog, the concentration ability will go up to 2400. In humans, only 1200, right? But for dogs, it will go up to 2400. So the same way, it starts with 300 and then it is moving to the medullary section. That means this is from cortical region to the medullary region. So whenever from cortex region to the medullary region something is going, then the concentration increases. Fine. So it becomes 1200. Then from again medullary part to the cortex it moves. As a result, its concentration decreases. So this is the most important part of the counter current multiplier. This is how it will keep on increasing its concentration from 300 to 1200 it will reach, right? So once the counter current multiplier, that means it will keep on multiplying like that and it will reach one maximum limit. Fine. So that will results into the maximum absorption. Now once this taken place, then the next part of this one is that counter current exchanger. So counter current exchanger, to understand that, again I am drawing the loop of Henle. Now inside this loop of Henle, you have the vasa recta. Okay, the vasa recta. Now what happens? The loop of Henle is flowing in this way, right? Whereas the vasa recta, it will flow in an opposite direction. Remember, that is why it is a counter current mechanism, because it is happening in an opposite way. So due to that reason, in the initial phase, in the multiplier phase, what is happening? From the descending limb, it will enter into the interstitial fluid. Then from the interstitial fluid, it will enter into the vasa recta, the peritubular capillaries, right? Then on the ascending side, again the same thing will happen. From the ascending side, it will just only the solutes will move from the ascending side to the interstitium, and interstitium it will move to the peritubular capillaries. Okay, can you able to follow? Yes. Then what happens here is that this peritubular capillaries or the vasa recta is permeable for both solute as well as the water. Unlike the loop of Henle, where one side is permeable for solute and another side is permeable for water, it is not so. It is permeable for both solute as well as water. Whichever the concentration is high, that will enter from that side, right? And remember, this all flow is taking place just because of the concentration gradient. Wherever the higher concentration is there and lower concentration is there, it is moving from higher concentration to the lower concentration side. Remember, all the physiological principles you need to remember in all the mechanisms. Now from here, the solute particles will come and it stays in the interstitial fluid and that will enter here, right? Whereas the water molecules, that will enter from the descending side, get accumulated in the interstitial fluid, that will enter in the vasa recta, which is running parallel towards them. That's all. So here also what happens, the entry level, the osmolality will be 300 milliosmoles, but the exit point, when you see, it will be 350 milliosmoles. Why so? It would have taken more amount of solutes and the water molecules from the medullary region, but why it is taking very less amount of the molecules and the including water as well as solutes. This things is called as, this phenomena is called as medullary washout. Okay, so medullary washout is nothing but it is a gradual loss of the water or the solute from the medullary region, right? The washout of the medullary gradient. So the gradual loss. So this is called as medullary washout. How this medullary washout is taking place? This medullary washout is taking place just because of the sluggish movement of the blood inside the vasa recta or the peritubular capillaries. That's all. Very simple. As it is moving slowly, so due to that reason, the concentration gradient is existing. So the solutes as well as the water will enter from the interstitial fluid into the vasa recta, and then it will keep on moving. So as it is permeable for both water as well as the sodium, so when it enters 300 and when it reaches the medulla it will become 1200, and again when it goes up it will keep on reducing its concentration, and then at the end when it goes out it will become 350. That means only 50 molecules, which includes the solutes as well as the water only, is removed from the medullary region, but not with a greater extent. So this is called as medullary washout region, right? So this mechanism is called as countercurrent exchanger mechanism. Okay, first we have seen the multiplier, how the 300 become 1200 I told you, right? Repeatedly you have to do that from 300 to 400, 400 to 500, 500 to 600, like that it will reach 1200, then it will become countercurrent exchanger, right? So in the countercurrent exchanger, next important part is that urea cycle. Urea cycle is another important part in the countercurrent exchanger. So what urea does is that this is the loop of Henle, right? And this is the distal collecting tubule, and it will go, and this is the collecting duct or the collecting tubule, right? Now what happens here, under the influence of the hormone, the urea transporter is open. As a result, the urea will diffuses from the collecting duct to the interstitium, right? Then from interstitium it will enter into the distal collecting tubule, the ascending limb of loop of Henle. Now when it enters, what is happening here? It will add the concentration to the existing concentration in the ascending limb, and ascending limb is permeable to the solute. So due to that reason, one side it is allowing the urea to enter into it, and another side it is sending the necessary molecules which is required for the body, such as sodium chloride. These ions are getting absorbed in the, it will move from the tubular fluid to the interstitium. So the urea, right, from the collecting tubule it will enter into the interstitial fluid, and from the interstitial fluid it will enter into the ascending limb. Whereas on the other hand, in the ascending limb, whatever the sodium or the chloride is present, that will move out to the interstitium, and then it will enter into the vasa recta. So the urea which enters here, that will added with the concentration of the sodium chloride. So that means what? The solute concentration is keep on building up here. So due to that reason, the important particle, that is the sodium chloride, which is maximum getting absorbed. It is ensuring the ions which is required for the body to be absorbed at the max instead of urea. So that is why whenever the concentration is getting decreased, the concentration is building up with the help of the urea recycling. So the urea will add. So for example, here it become 300, right? Now 100 molecules, if it moves out, then it will become 200. Now in this 200, what is happening? The urea that will come and it will add. So again it will become 300, right? So this 300, again another 100 molecules will move out. Now which molecules are moving out here? The sodium and chloride, which is required for the body, but not the urea. But how it is helping? The urea is helping for the movement of the sodium and chloride by adding it to the concentration in the ascending limb. So this is very, very important. So this is how the urea recycle is very, very important in the countercurrent exchanger system. Fine. Now let us enter into the, so I hope that you might be cleared now about the countercurrent mechanism. Please go through the literature and the books so that it will be much more clear to you. And then next will be the renin-angiotensin system or the renin-angiotensin-aldosterone system. Let us see the classical pathways. So why the renin-angiotensin system is an important? Because it is a critical regulator of blood volume, electrolyte balance, as well as the blood pressure in order to maintain the homeostasis. Now, the baroreceptor reflex, which responds for the short-term fluctuations in the decreased blood arterial pressure, right? Whereas the RAAS mechanism will take up this part when there is an acute and chronic alteration is required. So What are the compounds which includes in the RAS mechanism is that first one is the renin, then second is that angiotensin 2, and the third is that aldosterone. These are the three important components. And what are the stimulus is required for these components to be released? The first is the decreased renal BP, blood pressure. So decreased blood pressure means obviously the volume also, blood volume also reduced, right? Hypovolemia as well as hypotension. Then next will be the decreased salt delivery to the distal convoluted tubule. Why the distal convoluted tubule? Just recollect the juxtaglomerular apparatus, how it is the same, right? Where the macula densa is present, then granular cells. So that is why the salt delivery is important for the DCT. Then the beta adrenergic or the beta agonist drugs. So these are the stimulus for the RAS mechanism. And what are the organs involved in the RAS mechanism? The kidney, lung, systemic vasculature, adrenal cortex, and the brain. Then the rate limiting enzyme in the RAS mechanism is renin. If renin is secreted, then the mechanism will get activated, otherwise it won't, right? Now next important compound we have seen is that angiotensin 2. Angiotensin 2's main important function I already told, it is a potent vasoconstrictor, don't forget that. So it is a vasoconstrictor, then it helps in the secretion of the aldosterone from the adrenal cortex, from the zona glomerulosa, and then as a result it together causes increased sodium reabsorption, okay, with the help of a sodium hydrogen antiporter in the proximal convoluted tubule, as well as it increases the sympathetic outflow from the CNS and releases the vasopressin from the hypothalamus. Then coming to the aldosterone, where the synthesis and secretion it's primarily regulated by the angiotensin that we have seen in the earlier slide, and also ACTH and the extracellular potassium concentration, right? So these are all will causes the synthesis and secretion of the aldosterone from the adrenal cortex. Then the sodium reabsorption, the aldosterone which will causes the sodium reabsorption with the help of the ENAC. ENAC means epithelial sodium channel. Epithelial sodium channel is also called as ENAC. In the principal cells in the collecting tubules, with the help of this channel it reabsorbs the sodium, and it also activates the sodium potassium ATPase pump at the basolateral surface of the apical cells, fine. And then it causes the salt and water homeostasis by regulating the thirst mechanism as well as the salt appetite via mineralocorticoid receptors which is present in the brain, especially in the hypothalamus, this thirst center and all, right? So let us come to the classical mechanism of the RAS. So reduced blood flow or reduced blood volume, you can say hypovolemia or hypotension is the primary stimulus, right? That results into the decreased GFR because the blood volume also less and the blood pressure also less. Due to that reason, very less amount of blood will enter inside the glomerulus. Due to that reason, the renal perfusion will be decreased GFR will be there. As a result, the sodium content also will be reduced because the GFR is less. That means in the filtrate, how much sodium will be filtered? Less amount. So this sodium chloride, less sodium chloride will go to the distal convoluted tubule where it will make a contact with the afferent and efferent arteriole, right? Where it is having the JG cells, the JJ apparatus, where the macula densa will sense the lower sodium chloride concentration and it will give signals to the juxtaglomerular granular cells where it will have a already synthesized and stored the renin in a granular form, right? So this renin will be released, cleaved, the prorenin will be cleaved and the renin will be released. The activated renin will be released in the afferent arteriole. Now this afferent arteriole already having the angiotensinogen which is released by the liver. The angiotensinogen is synthesized in the liver, right? And it gets secreted into the systemic circulation, then it will reach the afferent arteriole, through the afferent arteriole which is the glomerulus, right? So when the renin releases, it will come and cleave the angiotensinogen into angiotensin 1 with the help of the ACE or the angiotensin converting enzyme from the lungs. The angiotensin 2, it will go and acts on the angiotensin 1 receptors, Okay, it will go and bind with the angiotensin 1 receptors. Otherwise, there is one more mechanism. The angiotensin 2 will directly convert it into angiotensin 3, where the angiotensin 3 will go and bind with the angiotensin 1 receptors, and then it produces increased sympathetic outflow, aldosterone synthesis and release, sodium retention, ADH release, and vasoconstriction that we have seen before. Right. Next, the last aspect of today's topic is the diuresis. That means what? The diuresis means increased water secretion. That is called as diuresis. So, anti-diuretic hormone, that means this is the hormone which inhibits the release of the water, right? It will causes the maximum reabsorption, retention of the water, right? The same way, diuresis means it will increase the secretion of the water. Okay, so more water will be excreted from the body, the urine formation volume will be increased, right? So there are the drugs which causes the diuresis is called as diuretics. Now based on the mechanism, these diuretics are classified into 5 types. One is the first, it blocks the carbonic anhydrase. As a result, there will be an increased sodium bicarbonate excretion. Example is that acetazolamide, where it acts on the proximal convoluted tubule. Then osmotic diuretics, it increases the water excretion. Example is mannitol, where it acts on the distal convoluted tubule and the thin ascending limb of loop of Henle. Then next mechanism is that it blocks the sodium potassium 2 chloride co-transporters channel, which increases the sodium potassium chloride excretion, as the name suggests. So the example is the loop diuretics. The best example is that furosemide, where it acts on the thick ascending limb. Then next, it blocks the sodium chloride transporter. As a result, there will be increased sodium chloride excretion. Example is thiazide diuretics. For the thiazide diuretic is hydrochlorothiazide, where it acts on the DCT. Then antagonizes aldosterone receptor or potassium sparing, you can say in many books you will find. So it increases sodium excretion and potassium retention. So example is that aldosterone antagonist is the spironolactone, which acts on the collecting duct. So some of the common clinical conditions which is associated with the today's topic is that urinary incontinence. That means the control of the renal system is stopped. As a result, there will be a leakage of the urine will takes place. So this urinary incontinence is of 4 types. One is that urgency incontinence, urinary incontinence. Next will be the stress, and the third is that functional, and the last one is overflow. So these are the 4 types of urinary incontinence. That means it is a loss of bladder control, that's all. Then polyuria, it is the increased urine output, whereas oliguria, decreased urine output. Dysuria, that is pain during micturition or the urination process. Hematuria, it is loss of blood in the presence of blood in the urine. Then hemoglobinuria, presence of hemoglobin in the urine. Then proteinuria, albuminuria, it is the presence of protein in the urine. So there is one more important terminology is that natriuresis. Uresis already I told increased release, right? So natri means it is sodium. So increased release of the sodium. So today what we have seen, the countercurrent exchanger mechanism, countercurrent multiplier system comes under the countercurrent mechanism together. Then the RAAS mechanism, then diuresis and diuresis classification, what are the common conditions involved in it, right? So with that we finishes today's topic. Thank you.